So correct and foremost important multiple sequence alignment is a key towards a good phylogenetic prediction. So they are highly correlated, this phylogenetic analysis and the quality of multiple sequence alignment. So if two sequences in an organism are very similar, they are assumed to be derived from common ancestor. So organisms where we can see lots of matches, we can expect that they have been evolved from some common ancestors. So a good multiple sequence alignment where we see a good number of matches, that can lead us to think of some common ancestry. So the sequence alignments reveal which positions are conserved from the ancestors. So for example, here we have two sequences AAG, AATC. On the other side, we have AAG, AGTT. So we see that there are two mismatches. If we align these sequences, we can come up with a consensus, and where we can see that position 1, 2, 3, and 4, AAGA, they are conserved. On fifth, we can have either A or G, and then we have a T which is conserved, and then in the last position, we can have either C or T. So in this way, we can have a rough idea, like since these two are highly correlated with one another, we see large number of matches, we can expect that they have been derived from a common ancestors where we get these two substitutions. The progressive multiple sequence alignment method is where we have seen earlier that it aligns the most similar pairs and then it adds the more distant pairs into that particular alignment, and this alignment is influenced by already similarities which are present in the similar pair of sequences. So this progressive alignment is a first method that has been used in most of those phylogenetic predictions where in order to generate that multiple sequence alignment, they rely on this method. But it does not correctly represent the evolutionary history of the changes which have been occurred. And there are alternatives. So most phylogenetic methods assume that each position in a sequence can change independently from the other position. So there is independence between different sequence positions. How can we resolve gaps in the alignment? So as far as the evolution is concerned, the gaps in the alignments, they represent mutations. There can be insertions, deletions. On one side, we see that there is some nucleotide or amino acid is being incorporated, but on the other side we don't see anything. We see a gap over there, so that it means on the second strand or sequence there is a deletion. So these gaps can also be representing some genetic rearrangements where we can, these chromosomes and the genes which are present in them, they rearrange or shuffle themselves. So in this way these gaps can incorporate those genetic rearrangements. So in the phylogenetic methods, gaps are treated in different ways, and most of these methods they ignore those gaps. And there is another alternative to treat these gaps is by using sequence similarity scores as the base for phylogenetic analysis. So how do those two sequences are similar to one another? So that can be used also, and that can be incorporated also in order to resolve these gaps. So instead of using alignment and trying to decide what happened at each position, we try to use these similarity scores. So the similarity scores based on scoring matrices with gap scores are used by distance methods. So whenever we have those sequence similarities, we can incorporate those gaps in it, and then we can take two sequences, we can compare them, and then we can calculate their distances. So that approach is actually embedded in those distance methods. So we see that correct and accurate phylogenetic analysis depends upon the quality of multiple sequence alignment, and multiple sequence alignment algorithms, whichever one is efficient, that is a good point for phylogenetic analysis.